3. Okay, so where we left off, a Dutch guy named Hugo de Vries and a couple of other scientists independently rediscovered Mendel's work. It had been gathering dust for about 35 years. It had been published in a small scientific journal that wasn't that widely read. Mendel supposedly sent a copy of his work to Charles Darwin, who was still alive at the time, but Darwin never read it because he wasn't very good at reading German. He could read it, but it took a lot of very slow and painful work with a dictionary for him to read German. So he never read Mendel's paper. De Vries and others started repeating Mendel's experiments in 1900, and they soon revealed that Mendel's basic laws applied to virtually all eukaryotes, but there were some more wrinkles to it. And when I talk about this, I'm going to illustrate this with case studies. Again, don't memorize the case studies. You don't have to know all of the details about cat fur coloration unless you decide you want to be a professional cat breeder, which I'm assuming not many of you are going to want to do. Learn the principles behind them. I'm not going to ask you exam questions about cats. I could, however, ask you exam questions about corn snakes or Labrador retrievers or corn plants or quite a lot of things. I'm going to ask you about species that you've never seen, but you'll be able to solve the problems if you understand the principles that underlie them. I've already mentioned this. Mendel's laws were worked out, and he was kind of lucky in this. The genes that Mendel studied happened to come in only 2 alleles. So we had purple flowered peas or white flowered peas, and purple is dominant to white. Tall peas or dwarf peas, and tall is dominant to dwarf. Alleles tend to come in 2s. In the real world, genes can have more than 2 alleles. They can be 3, they can be 4, there can be 5. The highest number I can think of is 26. There may be genes with more alleles than that. And an example that you can see in cats is that there are 3 alleles of the gene for dark base color in fur. We can abbreviate these as capital B, lowercase b, and lowercase b prime. That's B with a little tick mark next to it. Professional geneticists might abbreviate these B1, B2, B3. Doesn't really matter what symbol you use, but I'm going to use big B, and little b prime. Big B stands for the allele for black fur, and that's dominant to little b, the allele for a medium brown, and it's also dominant to little b prime, the allele for light brown fur. Medium brown is recessive to black, but medium brown is dominant to light brown. I should mention that cat breeders call medium brown chocolate, and they call light brown cinnamon. I guess if you're a cat breeder, it helps to give them fancy names. But here's what these cats look like. Up at the top left is a black cat. Down at the lower left is a medium brown cat. And at the right is a light brown cat, or black chocolate and cinnamon, if you'd rather use the fancy words. The black cat could be homozygous for the big B allele, or it could be heterozygous and have 1 big B and 1 little b, or it could be heterozygous and have 1 big B allele and 1 little b prime allele. we can't tell just by looking. We could maybe tell if we knew this cat's ancestry and knew the fur color of its parents and grandparents, we could maybe work out what alleles it's carrying. But just by looking at it, all we know is that it has one copy of the big B dominant black fur allele. The chocolate cat down at the lower left might be homozygous for little b, the medium brown allele, or it could be heterozygous for the medium brown and the light brown allele. It could have one little b and one little b prime allele. Again, we can't tell just by looking. And then finally, the only genotype that a light brown cat could have is to be homozygous for little b prime. That's the only way that that light brown allele is going to appear in the phenotype, because light brown is recessive to both black and dark brown. So yeah, that is a pretty common situation where you have more than two alleles, and this is an easy to see example with kitty cats. Probably a more famous example is one that was worked out in 1900 by Karl Landsteiner, who won the first Nobel Prize in medicine for his work because it made blood transfusions possible. Human blood, of course, is divided into blood groups or blood types. Your blood type can be type A, type B, type AB, or type O. I've got type O blood. Some of you, of course, probably have different blood types. And that particular blood type is controlled by a single gene with 3 alleles. And in this case, we have a case of what's called codominance. If you have an A allele and a B allele, neither one covers up the other, and your blood type is AB. If you have type O blood, O is recessive to both A and B. So if you have type O blood, the only genes that you could be carrying are 2 copies of the O allele. Your genotype would be OO. But if you have type A blood, you could have the AA genotype or the AO genotype. And if you have type B blood, you could have the BB genotype or the BO genotype. So there's 6 possible genotypes, which between them give you one of 4 possible blood types. You will actually work with this in an upcoming lab. I'm not sure if it's this week or next, but you've got a genetics lab that involves a kind of soap opera plot with a medical mystery that you have to solve using your knowledge of the inheritance of human blood groups. I should mention, by the way, that whether your blood type is positive or negative, that actually depends on a completely different gene. If you're, O positive or O negative or AB positive or whatever it is. I don't want to take the time to explain exactly how this works, but I think it does come up in lab. But what the genes actually do is the genes put various molecular markers on the outside of your blood cells, and your body reacts to whatever markers you don't have. So if you have type A blood and I fill you full of blood from somebody with type B blood, your body will attack that foreign blood because it doesn't recognize type B blood as its own. The same if you are type B and I transfuse type A blood into you, that causes very severe illness. And until blood typing was understood, blood transfusions were extremely dangerous because if you gave somebody the wrong blood type, you'd make them worse. I'm not going to go into how, but this is ultimately the reason why people with type AB blood can receive blood from anybody, and people with type O can give blood to anybody. If you are type O, you're a universal donor, which makes your blood very medically useful, because they can give it to people like, accident victims where there isn't time to figure out what their blood type is. But again, I think you will get more of that in lecture, so I'm going to move on. But yeah, that is a well-known case where we have more than 2 alleles. There's another phenomenon called incomplete dominance, and I'll show you this in kitty cats, but first let's look at snapdragons, popular garden flowers. And here some are. And it turns out that if you cross a red snapdragon with a white snapdragon, in this case, the offspring come out in between. They're all pink. Huh? Now, that's one of the things that Mendel didn't find when he crossed a purple flowered pea and a white flowered pea. The offspring were not all light purple. But here you have this in snapdragons. And it turns out that if you cross 2 pink snapdragons, what you get is 1 red and 1 white snapdragon for every 2 pinks. Instead of that 3-to-1 ratio, you get a 1-to-2-to-1 ratio. And the reason is that there are 2 alleles in this case. We can abbreviate them capital R and lowercase r. And the red allele is dominant, but it's incompletely dominant. Remember that a dominant allele covers up the effect of a recessive allele. An incompletely dominant allele covers up the effect of a recessive allele, but it doesn't do it completely. It does not completely mask the recessive allele. So in this particular species, heterozygotes are pink. So if you cross a red and a white snapdragon, the offspring are all pink. If you cross 2 pink snapdragons, they have the genotype big R, little r. You can plug this into a Punnett square. Each one of the parents passes on either a big R or a little r allele to each of its offspring, not both. So you end up with a quarter of your offspring getting a red allele from mom and a red allele from dad, and those will look red. And half the offspring get a red allele from one parent and a white allele from the other parent, and those end up pink. And a quarter of the offspring get a white allele from each parent, and those end up white. So that is a case of incomplete dominance. And here's a kitty cat example. We've talked about how, the base dark color of a cat's coat may be black or dark brown or light brown, or should I say black or dark brown or light brown? But there's lots of other genes that affect the look of a cat, and one of them is a gene for white spotting, and that's incompletely dominant to the allele for solid color. On the left, that's a black cat, because it would have, maybe big B, big B, big B, big B, or maybe big B, little b alleles, giving it that black color. But it has 2 copies of the spotting gene. So we'd write its genotype as maybe big B, big B, big S, big S. And the effect of that is to give the cat so many white spots that it now looks like a white cat with black spots. Okay, that is a white spotted cat on the left, but it's got so many white spots that they've all joined together, and the cat is almost entirely white. On the right, that would be a black cat with a genotype s. That cat doesn't have any white spots. The cat in the center is a heterozygote, and it has white spots, but it doesn't have many. A cat that's a heterozygote might only have, oh, we've got one. I can't find him right now, or I'd show you. He's almost completely black, except he's got one little white spot on his belly. We have another cat who's also asleep right now somewhere in the house, who is, they call it a tuxedo pattern. He's got a white stripe down his forehead and a white lower face and kind of a white patch on his chest that makes him look like he's wearing a black tuxedo with a white shirt. That's actually a fairly common pattern. Other cats that are heterozygous might only have a little white star on the chest, or they might have white feet and a white tip of the tail, but they're still mostly black. So that is incomplete dominance because the heterozygotes are kind of in between the phenotype of either homozygote. So we do get this in kitty cats as well as in snapdragons. Big concept here, one that's important for you to know, is that genes can influence what other genes do. They're not really acting in a vacuum. They're not acting completely independently of each other, but one gene can modify what another gene does to the phenotype. And in cats, a great example of this is a gene with two alleles called dense and dilute. Dense is dominant, but if a cat is homozygous for the dilute allele, that will take whatever the base color the cat has and lighten it. It will actually change the way that granules of pigments are laid down inside the hairs themselves. So what you're looking at is a black cat, but this is a black cat that is also homozygous for the dilute allele. The dilute allele does not make the cat a particular color. It takes whatever color the cat already is and lightens it a notch. So this is not a gene for a color. There's no gene for gray. There's a gene for black, and there's a gene for lighten it up. And we call this epistasis. And it happens all the time where you have several genes that affect the same trait. So for example, on the left, that is a black cat because it's homozygous, probably, for the dominant black allele. And on the right, that is a medium brown cat because it's homozygous for the medium brown allele. But both also have, they're also homozygous for the dilute allele of a different gene. And so they have the dilute phenotype. So their base colors of black and medium brown get diluted into gray and tan. Or if you're a cat breeder, they're called blue and lilac. The cat on the left is of a breed called Russian blue. Even though it doesn't really look blue to me, that's what breeders call it. And on the right, you have a lilac cat. Because it will lie like a rug all over your house, I suppose. Anyway, never mind the names, but this is a simple case of epistasis. Genes modify the effect of other genes to create a phenotype. And this applies to things like human skin. We tend to define ourselves as being black or white. I mean, especially if you're Michael Jackson, or if you're Paul McCartney, ebony and ivory, things like that. And certainly our divisions into races are the kind of things that kind of you know, create major social hangups. I presumably don't have to tell you this, but we're not like pea plants where the flowers are either they're purple or they're white and there's nothing in between. As you can see from the picture, human skin color comes in a very wide range. From, we're all basically tan, but some people are very, very light tan, and some people are extremely dark, and some people are in the middle. And the reason for this is that last I checked, this was a couple of years ago, there were at least 9 major genes, each of which have multiple alleles that affect our skin color. So the inheritance of human skin color doesn't quite work as simply as the inheritance of flower color in pea plants. We've got 9 different genes, and they all affect each other through epistasis. This is a very epistatic trait. And this is why human skin color does not neatly fall into these classes. We're not really black or white. We're not really ebony or ivory. We all fall at some point along a wide range, along a continuum. And this is just what you expect when you have genes, or when you have phenotypes that are governed by many genes working together and influencing each other. And this is true for a lot of things like your height, your build, possibly even intelligence. They have some genetic component to them, but it's not as simple as just saying there's one gene that makes you either tall or short, or one gene with alleles that either make you smart or stupid. A lot of traits are much more complicated than that because of the effects of epistasis. Kind of the flip side of epistasis is called pleiotropy. And in pleiotropy, instead of several genes affecting one trait in the phenotype, you have one gene with several effects on phenotype. And this is an absolutely adorable, if slightly weird looking kitty cat example. The cat that you're seeing belongs to a breed called Devon Rex. Now, a Rex cat is one that's got wavy fur. You may not, it's kind of hard to see on this picture. You'd have to pet the cat to really feel it. But Rex cats do in fact have wavy fur, not straight fur, which is straight is what most cats have. All the cats I think I've ever seen have straight fur. Devon Rex is recessive, and the non-Devon Rex allele is dominant. And if a cat is homozygous for the Devon Rex allele, the cat will have wavy fur, but it also has this very distinctive build where you have ears that are huge, they're set low, and the face is wide. Devon Rex cats are sometimes called pixie cats or fairy cats, or to my eyes, they look kind of like Yoda, or possibly Baby Yoda. Do 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 do, Baby Yoda. Do 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 do. Sorry about that. Right. So you have at least 3 major effects on phenotype: the texture of the fur, the width of the face, and the size and location of the ears. And they're all ultimately the result of inheriting just one gene. That's pleiotropy. One gene, several different traits. Epistasis, you'd have several different genes affecting one trait. Somewhat more poignant example of pleiotropy, some of the white cats that you see are white spotted, and they just have so many spots that they blend together into a predominantly white coloration. But you can have cats that are purely white. There's a couple of ways that can happen. But cats that are homozygous for one particular white fur allele are white all over, and they have blue eyes, and they're usually deaf. So white, blue-eyed cats frequently have deafness, and the fur color, the eye color, and the deafness all ultimately result from the fact that they're homozygous for one allele, right? One genotype has several different effects on the phenotype. I'll give you a dog example here. This dog's called a Merle. I'm not sure what, I guess that might be an Australian Shepherd. I don't remember off the bat what breed that is. But Merle is a coat pattern in which whatever the base color is, if you have the Merle gene, patches of that base color lighten up. So this dog's base color is black, but you can see that dog's got irregular black patches and then various patches of gray and even white on the body. For some reason, if you have dachshunds, this is not called Merle, this is called dapple. And I say this because I had a dappled dachshund when I was a kid. Maybe some of you have. You can see things like this in other breeds. I think there's Merle, Great Danes, and Merle and some hunting breeds like Catahoula hounds, if memory serves. So you see this in a fair number of dogs. Merle is governed by one gene with two alleles. Merle is dominant to non-Merle. The problem is if you're homozygous for the Merle allele with the genotype we can write big M, dogs with that genotype not only lack pigment, they're also usually blind and or deaf with abnormalities of the eyes and ears. You can see this unfortunate dog has small and malformed eyes. This dog is blind. And that would be another case of pleiotropy because the Merle allele doesn't only affect coat color, it affects development of the eyes and ears. This is why you should never crossbreed two Merle dogs because about 25% of the offspring would be homozygous for the Merle allele, and they would be born with defects of the eyes and ears, probably would not have a very good life. So if you're a dog breeder, it's not ethical to breed two Merles together because roughly a quarter of your offspring would have birth defects. So there, don't breed Merle dogs together. Yeah, if you breed Merles, it'll make you haggard. Merle, haggard. That's a joke. Okay, never mind. Somewhat more extreme example, we have to go back to kitty cats, of course, but this is a Manx cat. Manx is a breed of cats that's born with no tail. They have no tails. They also tend to have longer hind legs, which means that when they run, they look a little bit like, they tend to hop. They look a little bit like rabbits. I've even heard people call these cabbits because they look like they're half rabbit and half cat, not half rat, half rabbit and half cat, but they're not. They are cats. They just have alleles that cause tails not to develop. The tailless allele, which we can write capital T, is dominant to the tailed allele. So every cat that you see that does have a tail is homozygous recessive, has a little t genotype, and that's most cats. If you cross two of these Manx cats, you get two tailless kittens for every tailed kitten. And the reason is that a Manx cat with the big T genotype, a Manx cat that's homozygous for the big T allele, doesn't ever develop. It stops developing and it dies when it's still an embryo inside mommy's body. Oh, it's never born, at least not born alive. And we call this an embryonic lethal. Um, I'm sure there are human examples of this, although I can't think of any off the top of my head. Uh, there are certainly examples of this in other animals as well. You can have cases where a particular genotype disrupts the development of an embryo so much that the embryo is never born. It never hatches out of the egg, or it's never born alive outside of its mother's uterus. That happens. So Manx cats have the genotype big T, little t. They're heterozygous because they have to be. They're heterozygous because homozygotes don't exist, at least not as living adult cats. So you can, if you did this, you can solve this with a very simple Punnett square. A cross between two Manx cats would give you, on average, one big T cat for every two heterozygous cats, big T. For every one little t cat. That big T cat is never born. And that's why instead of getting a 3-to-1 ratio of tailed to tailless cats, you get a 2-to-1 ratio. So that can mess with Mendel's ratios if some of the possible offspring are never seen because they are embryonic lethal. Okay. Last thing I want to sock you with. We've been talking as if genes predetermine exactly what the phenotype is going to look like. That if you're a pea plant and you inherit 2 copies of the dominant purple allele, your flowers are going to be purple, and that's that. Or if you're a cat and you inherit 2 copies of the dominant black fur allele, your basic fur color is going to be black, and that's it. That's all there is to it. It's not that simple. And here's a case in cats. Here's another example of epistasis for you. For a cat to show any color at all, it has to have at least 1 copy of a color gene. We can represent the color allele as capital C, and that allows whatever color the cat has to be expressed. Color is dominant to albino, which we can write as little c. And if a cat is homozygous for the albino allele, it will not show any coloration at all. It will still have genes for black or brown, possibly genes for dilute or dense or whatever other colors we've got, but it will never show them because if it doesn't have at least one of those dominant color alleles, the cat will never show color, even if it has, no matter what other genes it's got. That's another beautiful example of epistasis for you, by the way. But that gene for color has multiple alleles. There's big C, there's little c. There's a third allele that's recessive to capital C and dominant to little c, and we abbreviate it CS. And if a cat is homozygous for CS, that cat will show color, but it will only show color on the parts of the cat that are cool. So think about this. If you have a cat that is homozygous for CS alleles, so it has the genotype little cs, that cat may show color, but it's only going to show it on the body parts that are cooler than the rest. So if you think about this, the parts of a cat that are cool tend to be the same as the parts on you that get cold, if you're outdoors and you're not well wrapped up. If you're out in a short and t-shirts and suddenly the day gets chilly, what gets cold first? Well, it's your nose, your ears, probably your feet, and your hands. And this is true for cats as well. The coolest parts of a cat's body are farthest away from the core of that cat, and they tend to be the cat's ears and face and paws and tail. And this is what we get. This is the Siamese pattern. The cat on the left is a black cat, and that cat might have the alleles big B, for all I know. But that cat only expresses those alleles on the cold parts of its body, the face, the ears. You can't see them in this picture, but the feet, the tail, and if it's a boy cat, it will express black color on the testicles as well. Sorry, my wife had a cat named 8 Ball for this exact reason. If you were, and people have done this, if you were to take that cat on the left and shave a patch of hair on its back and then strap an ice pack to the shaved part, you would get a really pissed off cat that would crap in your shoes. But you would also get the hair growing back in a darker color. You could actually put dark spots on this cat by shaving off the light colored fur and then keeping that skin where the hair is growing back, keeping that skin cold. I haven't seen this myself, but my wife grew up with a Siamese cat that spent a lot of time outdoors, and her Siamese cat's entire body got darker in the winter and lighter in the summer. The whole body produced more pigment when the whole cat was exposed to cold weather. In the summer, the fur came in lighter. The cat on the right, by the way, the base color of that cat is light brown. But because that cat is also homozygous for the little cs allele, that cat is only showing light brown color on the cool parts of its body, the face, the ears, and again, the feet and the tail, although you can't see those in that picture. The big reason why I'm talking about this is that the environment very much can affect the way that genes create the phenotype. Here's a non-cat example that you'll be seeing around here soon. These are hydrangea plants. They're native to Asia, but they're very popular in gardens. I can tell you my mom has a bunch of them in her front yard. Maybe you or maybe some of your moms do as well. And they come in blue or pink. I took this picture at one of the Kroger supermarkets in town, by the way. So I know the stores have them. I bought my wife one for Valentine's Day, so you could see these now if you were to, go to a store with a nice flower department. And the reason I bring these up is you can see that we have blue and pink hydrangeas. That's not a genetic difference. Blue is not dominant to pink or anything like that. What happens is that plants can only make blue pigment if the soil is slightly acid. If they're in more alkaline soil, that's the opposite of acid, the flowers come out pink. And you can actually make your hydrangeas change color by altering their soil, by putting, adding compounds to your soil that will make it more or less acid. The fancy name for this, by the way, is phenotypic plasticity. Plastic, before it meant the material, plastic just meant able to be shaped, which makes sense because plastic, the material, is plastic in that you can make a lot of shapes out of it. Plasticity just means the ability to be shaped, the ability to be altered. Phenotypic plasticity means that the phenotype can be altered depending on the environment, as is the case with Siamese cats as well. In lab and in this lecture, we look at single genes with simple and straightforward effects, like Mendel's peas. The truth is that practically all genes are going to show some degree of epistasis and pleiotropy And they're also going to be influenced by the environment that an organism lives in. This is true, human skin color is dependent in part on what genes you inherit. It's also dependent on how much time you spend in the sun, right? The environment affects how much pigment is in your skin at any given time. So do your genes. And you get this all over the freaking map, that a lot of traits are the product of genes and environment working together, sometimes in very complex ways. The basic Mendel's laws that we went over on Friday are sometimes called beanbag genetics, where each gene and each allele, each gene is responsible for exactly one trait, and each allele gives you only, one effect on that trait. There's only one gene for flower color, and it has two alleles, purple or white. That's useful, but it's a simplification. And in some cases, it is a massive oversimplification. And that's just something I feel is important to know, that genes are important, but in many cases, they don't really program what your final phenotype is going to be. They don't really work like that. Your final phenotype is a product of the environment as well, and the environment can affect what your genes do. And I seem to be going 1 minute over time. So, by the power vested in me by the state of Arkansas, I will stop the recording now.